I wanted to bring in the topic of the Cure Baby, Mississippi Cure Baby, that was presented last year at CROI, and we had another report from Deborah Prasad this year, and I wondered if Sharon, you could give us a little update on that.
Yeah, so Deborah first of all gave us an update on the Mississippi Baby. Now 24 months off treatment, no rebound virus, but actually found traces of virus in the baby cells. So I think there was a little controversy early on about was the Mississippi Baby indeed infected, but what she showed us I think confirms that baby was, yes. And this is truly a remission for the Mississippi Baby, or functional cure.
And can you explain that a little bit about finding that residual virus? Because they also found it in Timothy Brown.
Yeah.
So
that's right. In Timothy Brown, it was much lower levels. And what you can do when you find the virus using a very fancy test that can detect very low levels of virus. And anyone that works in the lab using this sort of test, what's called a PCR test, you're always worried, is this laboratory contamination, because the test is so sensitive. And I think with Timothy Brown, the jury is not out. Was this real or was this contamination? I think with the Mississippi Baby, it's a little different because they found much higher levels of persistent DNA or traces of virus in multiple cell types. And they've also done some sequencing and show that it's not a laboratory contaminant.
Okay.
So I think it's a little different. She then told us about this new baby from California, near Los Angeles.
Los Angeles.
And this baby was delivered actually shortly after CROI. So I think that was informed the doctors to perhaps to then start immediate treatment on this baby at 4 hours following delivery. Baby was confirmed as infected using the standard tools that paediatricians use, which means 2 positive tests. And in fact, the baby also had evidence of virus in their spinal fluid before they started treatment at 4 hours. Started full dose antiviral treatment, again what the Mississippi Baby had, and detected virus shortly after, but then no detectable virus in blood. But detectable virus in cells or DNA. Baby's been on treatment for 8 months and hasn't yet stopped treatment. So I think it's a little early to even say this baby is in remission or it's a functional cure.
I mean, it's one of the questions that we have in some of these studies is that we have to really stop treatment to see if it's valid.
Yeah, you need to stop treatment. And of course, we learned from the Boston patients, we stopped treatment, a lot of excitement at 8 and 15 weeks, but then the virus came back.
Can you expand upon that a little bit? Because that was a big report at this conference. It was reported earlier, but it was reported again here.
Yeah, so the Boston patients were 2 men who had HIV, were on antiviral therapy and had a lymphoma and needed a bone marrow transplant for their lymphoma. And just like the Berlin patient, they received a transplant. But the difference here was, first, that the bone marrow that they received was from a regular donor. Nothing special about being resistant to HIV, which is different to the Berlin patient.
Right, and people get that confused, I think.
Yeah, the Berlin patient received a unique type of donor bone marrow that came from someone that was naturally resistant to HIV. The Boston patients received a bone marrow that was not resistant. The Boston patients also didn't have, mega irradiation therapy. They had sort of modified irradiation therapy. And but the Boston patients stayed on their anti-HIV drugs, antivirals, during the transplant and for 2 and 4 years after the transplant. And what the investigators, Tim Henrich and his team, found was that when they measured HIV in cells, which is the HIV DNA test, within 6 months that was cleared from the Boston patients. They tried to grow virus. They couldn't grow virus. They looked in one patient in the gastrointestinal tract, couldn't find virus. So after, I think it was 2 and 4 years on treatment, they did the ultimate test, which is stopping antiviral drugs and seeing if the virus came back. And there was a short window where things looked very exciting because it appeared that these patients were in remission. But as we've learnt before with HIV, there's always these unexpected findings, and the virus is tricky, and the 2 patients eventually did rebound at 8 and 15 weeks after stopping treatment. Now that is much longer than we normally see viral rebound. We normally see, when people stop treatment, the virus normally takes about 3 weeks to come back. So it took longer for the Boston patients, and we think that's a indirect measure of how much virus was left. If you've got a tiny amount of virus, it may take longer to come back.
Okay, so that in itself is an advance.
Yeah. I think that was a big advance, knowing that, and also, but the Boston patients are also a little different to regular patients who might stop their drugs, and that is they had a new immune system from their transplant, and the new immune system had never seen HIV before.
Okay.
So when the virus did come back, it was like a primary infection, and the virus went from undetectable to very high levels very quickly.
Yeah, interesting.
And that's probably because the Boston patients were effectively like patients that had never been infected before.
And do you know where Heinrich is going with this research? Do you know what his next steps are?
Yeah, I think he's really going to dissect the immunology very, very carefully. You know, we always think about T cells as being the major cell involved in keeping the virus under control. But there are many other immune cells that could be important, one particularly called natural killer cells. And maybe it's their immune footprint, or what we call HLA type, that was really relevant. So I think he's going to go into a lot of detail around the immunology, and also the virology. If you look at you can sequence the virus that popped back up. Where did it come from? To try and find out, where did that virus come from? Why couldn't we find it in the blood using our routine tests?
Are they going to look at other patients?
Yeah, I think everyone around the world is trying to find their patients. We have a patient, in fact, who received a allogeneic transplant under continuous antiretrovirals in that patient. But I think case of the Mississippi baby, cases of the Boston patients stimulate a lot of new ideas and new research. And these are rare cases, but it means everyone goes home and starts looking for them in their clinic. And if we can characterise more of these cases, I think we're going to learn a lot more.
So I think one of the questions that comes up around these transplant issues is that these are patients that are very sick with some kind of malignancy. So what is the next step beyond studying those particular types of patients? Or do we know?
Yeah, I mean, I think there are different ways to, there are different ways that you can clear out T cells than just by transplantation. You can use some immunomodulatory treatments. And that could be of interest. I think developing animal models where you can mimic this, and there is some work going on in non-human primates, which allows you to manipulate the system a lot more easily, and they may give us clues. Because you're right, we're looking in a population of people who have underlying lymphoma and underlying immune defects.
Yeah, and so I guess this kind of highlights that we're just still really very early in understanding this whole platform, this whole sort of pathway towards looking at remission.
Yeah, I think many investigators I think clinical trials and careful observational studies are really important because they generate new ideas and new lines of thinking. But I think all of us just keep coming back to the fact that we need to understand the basic biology a lot more to really tackle the problem and get new targets and approaches.
Yeah, the early answers lead to more questions.